We've seen that metabolic reactions include anabolism and catabolism. There's so many of these reactions that are taking place all of the time in our body, and they have to take place in a particular sequence. It is enzymes that control the rates of these metabolic reactions. Enzymes are catalysts, that is, they speed up the rate of the reaction. You could eat a bologna sandwich, and if there were no enzymes, that bologna sandwich would look exactly the same 50 years later in your stomach without enzymes to break it down. What enzymes do is lower what is called the activation energy. It's the energy needed for the reaction to take place. We can see reactions taking place if we heat things up. For example, if you add a lot of sugar to water, you can heat it up to get that sugar to dissolve. But our body cannot be heated up indefinitely, or we will die. So we use enzymes to speed up these chemical reactions. Enzymes are three-dimensional proteins, and their function is determined by their shape. They have extremely specific shapes. The site on the enzyme that is the functioning site is called the active site. Enzymes are recycled, they are not reused, so at the end of a reaction the enzyme is the same. It is extremely specific. The molecule that the enzyme acts on is called the substrate, and every single enzyme has its own substrate. This diagram here represents a metabolic pathway. The substrates, that is the molecule the enzyme acts on, are the boxes, and each enzyme is labeled A, B, C, etc. So what happens here is substrate 1 is acted upon by enzyme A. Maybe a bond is broken or a bond is formed, and it becomes substrate 2. That substrate is acted upon by enzyme B, and it becomes substrate 3. So very small changes occur to this molecule by the action of the enzymes, and finally we reach the product. This product it's the end of a series of reactions, and the product itself may stop the reaction in a process called negative feedback. We can identify enzymes by the ending, the ASE ending, just as sugars had an OSE ending, such as glucose, sucrose, maltose. Enzymes have an ASE ending, and we know what substrate it works on. For example, on the milk sugar lactose, the enzyme that breaks that down would be lactase. Sucrose, table sugar, would be broken down by sucrase. So here we are back to this metabolic pathway. The product here is going to regulate, it may regulate the rate of the reaction. For example, once the cell has enough product, it does not want to continue to make that product. It only makes enough product that's needed. So the product itself can go back to the beginning step, that rate limiting step of enzyme A, the very first enzyme, and basically shut that enzyme off. It's a physical reaction. The product will physically interact with that enzyme and turn it off. Once all of that product is used up in the cell and more product is needed, that product disconnects from the enzyme, and the enzyme A now functions as the first step in that biochemical pathway, and the product will be made. So it's a nice way to shut down that whole product, that whole metabolic pathway without producing too much product. Enzymes may need what are called cofactors and coenzymes in order to be functional. So an enzyme may be basically an immature enzyme until it connects to its cofactor. Some cofactors, examples would be iron or zinc or copper. It's non-protein, and it activates the enzyme. Coenzymes are similar to cofactors, however they're organic. So some of our vitamins that we consume are coenzymes. We must consume these vitamins. We cannot make them, and we will die without consuming our vitamins. Enzymes are proteins, and proteins are affected by a variety of things in the body. For example, heat. You cannot overheat the body too much or your enzymes will denature. This is one reason that newborn babies are so heat sensitive. Their body temperature cannot go up too high or their proteins are denatured. Once a protein is denatured, that is, it goes from its quaternary state all the way back to its primary state, primary structure. That cannot be reversed. So the denaturing basically destroys the enzyme. Other factors that alter the enzyme would be changes in pH in the body, different chemicals, radiation, electricity. So here's one of the many reasons our body must maintain homeostasis is to keep the enzymes healthy and active.